Good afternoon, everyone. I am Suzanne Marselis, and I'm going to tell you about my research that is about exploring the potential of GEDI-derived canopy structure for mapping tree species diversity in the tropics. First, I want to make sure that I acknowledge everyone, because a lot of different people have contributed their data to this research, and without these people and without the organizations and the institutions that are collecting this data, this research would not have been possible. So thank you very much for contributing your data. All right, so, well, mapping tree species diversity at a high space resolution is, of course, quite relevant in order to enable effective conservation and biodiversity management, but also to allow for a better understanding of relationships between forest composition and productivity across different scales. So, traditionally, tree species diversity maps might be based on the extrapolation of field inventory information, or the combination of environmental variables with field-derived tree species diversity information, and then predicting how many different tree species there are in different areas of the world. My goal is to evaluate if we can use full-waveform LiDAR data for mapping tree species diversity, because this could be very relevant in light of the GEDI mission, which is going to provide a lot of these full-waveform LiDAR measurements across the world that we might be able to use if a relationship exists between the number of different tree species and the vertical canopy structure. So, let's see. Previously, it had been hypothesized that canopy height might relate to tree species diversity, because you might be able to imagine that a canopy height gives you information about the volume, so basically about the available niche space that there is for different species to grow. And it has been shown that there is a relationship between canopy height and tree species diversity, or the number of different tree species, even across the world, but it's not like very strong. So I hypothesized that if we would include information about the vertical canopy structure, we might be able to explain more of the variation in tree species diversity, because now we don't only have information about how much space there is, but also about the occupation of that space along the vertical axis. So I'm going to show you results of actually two different studies that I pulled together in this presentation. First, I'm going to show you that we explored the relationship between this just canopy height and tree species diversity in Gabon, and then we include vertical canopy structure and see how that relates to tree species diversity. Subsequently, we see how this relation holds when we look at it across Central Africa and eventually across the entire tropics. And well, I already said that it would be relevant in the context of GEDI, so it's a bit double, but all right. So basically the data that we have available is like two different data sets. So there's field data inventory. So for the field data, we use information about the different tree species. So just imagine like a field plot and you know which trees are there, and then you have identified the species. So now you can calculate how many different tree species there are within a certain plot. So it's tree species richness, basically. And then we have LiDAR waveforms. So from the LiDAR waveforms, they are either collected by the ELVIS, NASA's airborne instrument, or they are simulated GEDI waveforms from other airborne LiDAR systems. And we basically have multiple or quite a lot of waveforms within each plot, and we just calculate the average within one plot to be representative of the canopy structure within that plot. So the information that we use is we use canopy height derived from the waveform, and then we use this plant area index profile that Fabian was also talking about. So basically it gives you information on how much plant material there is in different vertical bins along the forest vertical axis. All right, so in Gabon, these are data from 3 different study sites and just relating canopy height to the number of different tree species. In this occasion, you can see that just canopy height alone explains about 44% of the variation in tree species richness. Now what we do is we include in this model also the vertical structure metrics that we have derived from the waveform. And this is a plot showing you measured versus predicted number of trees within a quarter hectare plot. And you can see that this relationship is definitely improved, and with this model we're able to explain up to about 70% of the variation in tree species richness. Now there is, as you all may know, the number of different tree species per plot. If you would change the size of that plot, then this number of different tree species is not going to change linearly with the plot size. This is called the species area relationship that most of you might be familiar with. So what I did is actually explore this relationship at different plot sizes, because I figured that if the canopy structure is rather similar across different plot sizes, but the number of different tree species doesn't change linearly, then there must be some more optimal resolution at which this model might work or not. So that does seem to be the case. These are the results from these models. So on the x-axis there's like different plot sizes, and then on the middle panel there's like the R-square of the model. So you see that that quarter actor resolution, the one that the scatter plot is from, is like the better resolution. And if you go either larger or smaller, it gets a little bit or a lot worse. All right, so basically what we can tell now is that yes, vertical canopy structure does explain more of this variation in tree species richness than just using canopy height alone, and it is promising enough to study further and see how this could hold up across larger areas or even across the tropics. So first what we did was we looked at just Central Africa, including not just only data from Gabon, but also from the DRC. And here's a scatter plot again of the measured versus the predicted results. And if we look at the model specifics, you can see that the top line is the same line we were looking at earlier. It's from Gabon, like the light blue one. So, and the darker blue one, which is the bottom one, is the one from Central Africa. So you can see that we lose a little bit of predictive power when we make this area, the spatial area, larger, but we're still able to build a relatively good model. Now, we want to see what happens when we go all around the tropics. In a way, you would expect that it shouldn't really work, because what we know from biogeographical history is that a lot of these regions, they develop differently. So, for example, we know that in Borneo there is like super tall trees, but the most diverse areas are in South America. So it wouldn't be surprising if it doesn't work like perfectly well. Oh yeah, so we now have data from 15 different study sites. Oh, let's go back across 4 of this continent. And each of the data still has like this field data, this number of different tree species per plot. We're still looking at 3 different plot sizes, and we have either Elvis LiDAR waveforms or simulated GEDI waveforms. So this is just to show you what the relation looks like between this canopy height and the species richness. So canopy height is on the left side, and total plant area index is on the right side. So you can see, for example, in the top, oh, here, yeah. So like different canopy heights may lead to vastly different numbers of species. Like this is all the same canopy height, but the number of species might be quite different. Whereas if you move more down, here you basically have less of these outliers, but you also have a lot more just like scatter around a more straight line between the relationship. So if we look at, if we can build a model using canopy structure and canopy height to explain this across the tropics, then, all right, well, it does something. It is definitely a significant model. We can explain up to about 40% of the variation in tree species richness, just throw in all the data together. And in that situation, we also see that actually like the smaller plot size is the better plot size. This is very advantageous because that happens to be pretty close to the GEDI footprint, so that worked out. Well, there's obviously a bunch of limitations on this study. One is that, you know, even though we have 15 data sets across the tropics, they're not particularly completely representative of all the variation that there is. And this should definitely be taken into account when we're looking at extrapolating any of this for predicting real tree species richness across the tropics. Also, I was doing this analysis at different plot sizes, but basically it depended on the characteristics of the original field data sets, whether I would be able to use the plot at different sizes. Because, for example, if a plot has a stem map, I can chop the plot into smaller pieces and do the analysis at multiple sizes. But if the plot is just like one size, and we know all the trees that are in there, but we don't know where they are, then the plot is only usable at one size, which makes it really difficult to really compare what resolution would work better or worse. Of course, there is the analysis or the goodness of the analysis is kind of dependent too on the quality of the data, and what happens, and especially in the tropics, is it can be really difficult to identify all the trees that are in a plot, because as most of us might have been in a tropical forest and just seen that, you don't even know what you're looking at. So I tried to exclude like plots that had a lot of trees that were unidentified, but there's still probably, it adds some noise into the model. Then, okay, now what did we learn and how can we use this on GEDI? Well, I think that if we want, we could just apply the smallest resolution model. So this is like a 25 by 25 meter plot. We could apply it to incoming GEDI waveforms, and we could basically make footprint level predictions of tree species richness in Gabon. And then we could compare them to the ones that we have from wall-to-wall areas of airborne LiDAR data. But because GEDI is a sampling mission, like Ralph explained in the morning, you only would be able to get like footprint level predictions, and there will be like gaps in between all your predictions. It is more difficult to extrapolate a prediction of tree species richness to a different area because of this nonlinear relationship between number of species and the area, and that also changes by different regions that you might be in. So footprint level predictions it is. Then, now what does it mean for the whole tropics? Okay, the model might explain 40% of the variation in tree species richness. And this is a model at the GEDI footprint level. So we could apply it to GEDI footprints, but if we would like to make a more reliable map of pantropical species richness just based on GEDI data, it would be better if we could develop a set of regional models that would in the end cover the entire tropics. But this would be contingent of a lot more calibration and validation data that is currently not available for me to use. And then, ideally in the end, I guess the best way to go would be to use data fusion. Data fusion could either allow for wall-to-wall mapping by fusing it with, let's say, TandemX or other type of radar data that gives you a wall-to-wall structure map that you could use to apply your models on, or you could fuse it with spectral information, hoping that maybe the spectral data will explain a different portion of the variation of your tree species diversity and improve the accuracy of the models. Or you might just go with a different approach and use gridded canopy structure information from GEDI in, let's say, a framework that is using environmental data and species richness reference data to predict tree species richness across the tropics. Oh wait, conclusion. Canopy structure can explain a limited percentage of tree species richness across the tropics, and GEDI will provide information that will be useful for this, but there's definitely more research needed for this to be operationalized.